Hello again. That was my mom and dad. So they were coming in to see the baby, and it ended up being my husband and kids came in at the same time. So we had a little visit, and now it's a little quieter. So I'm going to try and do some more of your lesson. Um, we got to starting to do this test cross for the dihybrid cross. Um, so we said that in order to do the test cross, you need to cross the individual in question with an individual who's homozygous recessive for both traits. Okay, so this is the genotype that you'd be mating with, and there's two possible genotypes, and we're trying to figure out by looking at what offspring they have, we can look back at the possibilities and figure out um, which genotype the other parent must be. So we have two possibilities. We have big P, um, oops, big P, move that out of the way. Wrinkled, so it would be little r. Or we have big P, little r. And both of them are going to be mated with the one that's homozygous recessive. All right, so if we have big P, little r, and we have little p, little r, then here's our Punnett square. Oop, my boxes are not very even, but it's kind of awkward. The possible gametes for this one is going to be big P, little r. That's all they really can give. Oops, what's going on? Cranky thing. Big P, little r. Okay, so it's all the same. So this one could be fairly quick, really, because we know once you see that the gamete possibilities are all the same, you can really look at just a couple of the boxes. So the only possibilities for the one that you're testing it with are little p, little r for everyone. Okay, so all the babies should be big, can't even see that. Here, I'll use a bigger box. Big P, little r, little r. Okay, so that means, oop, what is going on with you? All would be, big P is purple. All are purple. It's not letting me write. All are purple with wrinkled. Okay. Now, if it was heterozygous purple, then big P, little r, little r. Okay, so we'd have the possibilities are big P, little r for these gametes, big P, little r, or little p, little r. Okay, the only ones that these can give are little p, little r, right? So you could fill in the whole thing, right? Or you could say, oh, well, big P, little r, little r. That's going to be the same all the way across. It's going to be the same for this row. So you'd have 8 of them that are big P, little r, and you'd have 8 that are little p, little r, little r. Okay, so you could say 50% one, 50% the other, or you could say 2 to 2, or you could say 8 to 8. However, sense to you is fine. Okay, so we could say 1 to 1. We could say 50 big P, so purple, wrinkled. We could even say it in percent. 50% are purple and wrinkled. 50% are white and wrinkled. Okay, so then it says underneath, if in an actual cross between this unknown plant and a plant with white flowers and wrinkled seeds. 3 of the 7 produced had white flowers and wrinkled seeds. What was the genotype of the unknown parent plant? So you'd look at the possible phenotypes, and it says this one, the one on the left, that's homozygous dominant, would be all purple, wrinkled. The one on the right, that's heterozygous dominant, has 50-50 chance of being purple or white flowers. Okay, they're all wrinkled, but 50-50 purple or white. So we know, given that this actual cross produced some that had white flowers, that the genotype must have been heterozygous dominant. Oop, okay, so I think that in those questions that I assigned, number 2 and 3, page 544, I think one of them is a test cross. I'm hoping it is. If not, then we'll be getting you another sample after. Okay. So, so far we've been talking mainly about complete dominance and fairly straightforward traits. Okay, based on Mendel's findings. So this one is just kind of summarizing the fact that his findings were made between 1853 and 1861, when nothing was really known about chromosomes or genes or meiosis. So early on, he figured, okay, each trait is governed by 2 factors. Okay, and we know now that that refers to the 2 genes, one from our mom, one from our dad. Okay, the factors segregate, so each gamete has 1 factor. Inheritance of the factors for one gene or trait does not affect the inheritance or factors for another trait. So they're passed on independently of another. That's what the dihybrid cross was showing us, independent or law of segregation. Okay, his findings were fairly well unnoticed until the early 1900s. So like a lot of scientists, his work didn't really wasn't famous when he was alive, but certainly has been recognized since. Okay, in 1902, Walter Sutton and Theodore Boveri studied the phases of meiosis and noted 3 main observations: that chromosomes occur in pairs, that these pairs segregate or separate during anaphase 1, chromosomes align independently along the equator of the cell. Okay, so remember when they line up, the mother and father copies can be on either side. It's very random how they are lining up. So these 3 things form the basis of the chromosome theory of inheritance, which says Mendel's factors or genes are carried on chromosomes. The segregation and independent assortment of chromosomes during meiosis accounts for the patterns of inheritance that we see. Okay, now this theory will actually account for patterns that don't follow Mendel's laws. So there are more complex patterns that we're going to be looking at, and Mendel's ratios wouldn't explain them because his dealt with complete dominance, basically traits that have either a dominant or a recessive form. It doesn't deal with ones that are governed by more than 1 gene or have different sharing of information. So we're going to start looking at those. So you should have an idea of the importance of these different people and their contributions to understanding inheritance. Okay, so yeah, there's a few names you're going to have to know and recognize, and kind of on a test it'd probably be more like match the person with what their findings were, or like multiple choice, or maybe even a fill in the blank. But to be a fill in the blank, you'd have to be someone or something that you should know fairly well. Okay, so these are just showing the 3 things that there are 2 factors, and each one is on a chromosome. Okay, so these are Mendel's factors, what he knew as factors. We have two, one from each chromosome, so a mother copy and a father copy, that they separate independently, so they're pulled apart during meiosis, and that the way or direction that they're pulled apart is random. So you could have the father copy of one and the mother copy of the other on one side being pulled apart, or you could have um, the both the father copies on the one side being pulled apart from the other. So it's very random. If you remember when we talked about how they line up, very random. And the more chromosomes you have lining up different ways, the more variety you can have in the offspring. Okay, so along came a fella named Thomas Morgan, and he used to study fruit flies. If you take genetics at university, you will probably get to study fruit flies, and it's kind of it's kind of neat, it's kind of fun, but it's also kind of gross because if they get out, then you've got fruit flies on the whole floor. I can remember we had fruit flies everywhere. Um, Anyhow, they're used a lot when you're doing genetics because it's easy to um, make mate them, and they have a short reproductive cycle, um, and they have a lot of different traits that you can look at, which is kind of unbelievable because they're so tiny, but it is pretty cool. So anyhow, Morgan noticed a unique pattern of inheritance when he crossed two red-eyed parents. Um, to him, he figured, I guess, they'd have um, raw red-eyed babies, but he obtained a white-eyed male. He expected the red eye to be dominant and white eye recessive, like in normal crosses. But when he crossed a red-eyed female um, offspring from the white-eyed male, what am I saying? He crossed a red-eyed female offspring of the white-eyed male with a normal red-eyed male. Um, what would he expect to see in offspring if it was a normal monohybrid cross? Okay, so let's have a look. He crossed a red-eyed female offspring, so he figured that it was big R, little r, with a normal red-eyed male. So it could be big R, little r by big R, or big R, little r by big R, little r. Okay, so you get a big R and a little r by big R and big R. In this case, they should all be red. Okay, this could be big R and little r, big R and little r, big R, big R, big R, little r, big R, big R. So in this case, he'd have a quarter of them be white-eyed if it was according to Mendel's laws of normal monohybrid cross. But he found that all the females were red-eyed and half the males were red-eyed and half were white. Okay, which suggested that eye color was somehow linked to sex, to the X chromosome. Morgan's studies on fruit fly eye color and subsequent work showed some traits are actually linked to certain chromosomes, like fruit fly eye color is linked to the X chromosome. Traits on the same chromosome are usually inherited together. They're usually linked and will segregate together. Genes located farther apart on the chromosomes can sometimes segregate separate from one another because of crossing over. So if you remember when we learned about meiosis, we talked about the possibility of crossing over during synapsis in prophase 1. So in that case, because of this possibility, they had to kind of rephrase Mendel's law of independent assortment. Instead of just saying that genes located on different chromosomes will assort independently, they had to include a stipulation for if crossing over does not take place. Okay, because if crossing over does take place, then sometimes the regions can assort together. Okay, so the gene chromosome theory. Genes exist at specific sites or loci, locus, plural, arranged in a linear fashion along the chromosomes. Knowing the fact that the genes were aligned this way helps them to determine that these genes, that some that are farther apart are more likely to segregate. So the more they learned about chromosomes and how genes are positioned along it, the more they could understand what was happening in the pattern of inheritance. Okay, so what they're saying is if they are farther apart, then there is a bigger chance that, for instance, this region and this region could be exchanged. So this part could be segregated from that part, and this part be segregated from that part. Okay. All right, so I think I'll stop there and start another recording. I'm not sure how far along you are in the class. I think now would be a good time to have a visitor. So if you could get your notebooks out, we're going to have the next part be about this visitor.